for joining us this evening. For those of you that don't know me, I'm Deborah Montague and I'm the chair of Out Positive UK. And I'm delighted that we've got three distinguished doctors here with us this evening. Professor Sanjay Popat, Ross Camidge and Dr. Melanie MacKean. They are hoping to be able to answer all the questions that you've been kindly sending in over the last 2 weeks. I'm especially grateful for them joining us this evening because they've actually all literally just come off another virtual meeting. I won't ask them to introduce themselves this evening because, as you can see, you can access their biographies on the platform, and I would like us to make the best use of the time that we have. So I will be directing the questions to the various presenters, but if anyone has any particular strong feelings, then they're able to jump in. So shall we get started? So I'd like to start with Professor Popat, and I think an easy one for you here. So we still hear of non-smokers who have been diagnosed at stage 4, who've been given quite a bleak prognosis and started on chemo, only to be told later that they have a treatable mutation. Wouldn't it be better not to start the chemo for non-smokers until the biopsy results are known?
Yeah, I agree, actually. Deborah, that's the way it should really be happening. You know, the treatment landscape has changed over the last 15 years or so. So really now, if you have what looks like non-small cell lung cancer and it looks metastatic, the number one step is to get the molecular profile. And obviously ALK is one of the key molecular results we have to get. And knowing that the patient is a never smoker means there's a very high probability of there being some sort of actionable mutation. May not be ALK, maybe something else. And starting on an ALK inhibitor as the first treatment is the ideal way of doing things. In the NHS, it's definitely the best way of doing things, because if you don't, it limits the treatment options that we have a little bit. We don't have as much flexibility to do what we would like because of the way NICE has made their decisions on what you start off with, then determines what additional treatments you have. So yes, we should. Having said all of that, you know, we do see patients who, you know, we diagnose with the disease and actually they're in trouble. And, you know, they're really not very well. And I may know in the back of my mind that the ALK result or the EGFR result or the molecular profiling result is going to take 2.5, 3 weeks, 4 weeks to turn around because of various local issues. That patient may not be able to wait for treatment for that period of time. So on rare occasions, we do start treatment with chemotherapy, but that's not really the rule. And I'm sure my colleagues online will agree. Feel free to chip in.
Okay, thank you. Thank you very much for that. Dr. Camidge, one for you. Are crizotinib and ceritinib now redundant? Well, I ask because we now have members who have actually been on these two drugs for many years.
So there's a difference between saying if you're on something and you're doing well, you know, just because it's bad for other people, you know, you should walk away from it. No, if you know, the bird in the hand is worth two in the bush. However, if you walk through the door today, are either of those drugs going to be ones I would start someone on? And the answer is no. I think ceritinib, apart from for the people who are on it and doing well, probably needs to be mothballed. Crizotinib still has some role, even if it's not the drug you start on, because it's a bit like a Swiss Army knife. It's got a few other sort of tools in it. So it's also a MET inhibitor. And at least on this side of the Atlantic, sometimes we add that in when we've proven that MET is a mechanism of acquired resistance.
Okay, thank you. And just don't know whether Dr. MacKean rather, or Dr. Popat want to add anything from this side of the Atlantic?
I just totally agree with that statement. If you're on crizotinib and doing well, why change it? And exactly that. We've got patients 5 years out on crizotinib who are doing well. Absolutely wouldn't change it for the world. We've got lots of options after that. So, you know, if it's working for you, that's brilliant.
Excellent. Thank you very much. I have a question now for you, for Dr. MacKean, and possibly for you, Dr. Popat. Alectinib and brigatinib have approval for first-line use in the UK. How do you decide which one to prescribe?
Well, this is exactly what we've been discussing in our virtual meeting prior to this. And the truth is, there is no strong opinion either way. There are pros and cons to both. It's wonderful that we've got so much choice. And the truth is, there'll be patients who can't tolerate one who we can quickly change onto the other. We're allowed to do that if it's intolerable or side effects. So that's wonderful. We've got two great options first line. There are some, practical things, literally things like the number of tablets that can sway you one way or the other. But honestly, we are open to both. It's a wonderful situation to be in, actually. Sanjay, any thoughts on that?
Yeah, thanks. I mean, I think that's one of the really important questions that we all have, and I go to many roundtables and have many discussions around this, and most of them end up with the same sort of conclusion, which is actually there probably isn't a huge amount of difference between the two drugs. They behave slightly differently. They have slightly different side effect profiles. They have a different tablet number that you have to take. The data on brigatinib allows us to use it in patients that we may have started chemotherapy before we knew the ALK result, and but actually I don't really see any major major demarcations. I mean, it's not like crizotinib and ceritinib, which are completely two different ball games. These are actually two very, very similar drugs. There are more similarities than there are differences. And Ross, I don't know what your thoughts are.
Yeah, I mean, just as we said in the last meeting before this one of the things that certainly influences my decision making is knowing what my options for plan B are. If everything is going to lead me to lorlatinib, then it really is a kind of, you pays your money and you takes your choice between alectinib and brigatinib. If you have more flexibility, and you may not, but if you have more flexibility in terms of being able to slot brigatinib in between alectinib and lorlatinib, then that, for my money, would be the best option. But that's actually probably not on label in the UK.
No, and actually, saying that, Dr. Camidge, the FDA recently approved lorlatinib for first-line approval now in the US. So you have a choice of 3 first-line drugs now, and I know it's been applied for with NICE over here.
Yeah.
How do you choose between 3?
Well, so lorlatinib I actually find easier, and I find lorlatinib actually takes the debate in a really interesting direction. So lorlatinib probably, when the data is mature, will have the longest, what they call progression-free survival, the time before the cancer starts to grow, compared to these other drugs, partly because it's just awesome at getting into the brain and it protects your brain. But it brings with it quite a lot of baggage in terms of side effects. And one of the things is, people, this is the good news, that, people with ALK-positive lung cancer can now live so long that we don't have to assume that progression-free survival equals overall survival. That first progression-free survival is only a tiny fraction of the total journey. And so maybe we can push a somewhat more toxic drug to say, well, hold on, maybe that's going to be my plan B, and I'll spend more time on a less toxic drug. Okay, I'll have to make a treatment decision a few months earlier, but so what? So at least in my practice, and I'd say the US it's not exactly split 50-50. I would say it's probably 70-30, with the 30 being they would go for lorlatinib, and the 70% saying, I'm good with alectinib, guys, keep lorlatinib till later.
That's very interesting. Thank you very much indeed. Thank you. All right, Dr. MacKean, there's another question here. I'm on alectinib and the tumour in my lung has reduced in size. Why isn't my oncologist using radiation as well as alectinib to get rid of the cancer?
So I'm really pleased to hear that you're responding to treatment. That's wonderful. And it often depends. We talk about oligoprogression. So if we see one site has shrunk and then is beginning to grow again, that's when we would go in all guns blazing with radiotherapy as a targeted calm it down. If it's all still shrinking, then we wouldn't apply radiotherapy at that point, because there will be disease elsewhere if you're having it for an advanced stage disease. And whilst it's controlling that, we hang on, keep that bullet for later on when we need it. I don't know if Sanjay, would you feel the same?
Yeah, I mean, in general, we tend to use that sort of high dose targeted radiotherapy like cyberknife radiotherapy or stereotactic radiotherapy when you've got a cancerous lump which is shrunk down beautifully and then showing some signs of growth. There are very rare patients where they have a very small amount of tumor to start off with, and this is the real critical issue. They didn't have a large amount of tumor that they started off with. They may have only had one small lump in their lung, and that was it. And they were classified as having metastatic disease because, for example, they may have had 2 or 3 cancerous deposits in the brain. Now, that is extremely rare scenario to be in. Now, in that sort of scenario, you might want to make an argument to give the radiation to the lung if you've treated the brain with radiation and everything is shrinking down. But that is a very rare scenario. The vast majority of patients that we see routinely have got quite widespread disease at the time of diagnosis, and then most of it may have gone away, but still you'll have the fingerprints of the disease inside the body, but you may still see that lump there. I'm not sure that we really have the data that we should be consolidating with high dose radiation for those patients quite just yet. But I'm interested to hear Ross's thoughts.
So this is obviously an evolving paradigm. So we like the word oligo, meaning small. So we talked about oligo metastatic, small number of sites of disease when you walk through the door, like Sanjay just talked about. We've talked about a small number of sites growing when everything else is controlled. That was oligoprogression that Melanie mentioned. But there is this middle category of kind of like oligo-residual disease. So you can have had 50 deposits, 49 of them disappear and something's left. What about jumping in with radiotherapy there? So there's a little bit of data, not specifically in ALK, that consolidation radiotherapy can be beneficial. But of course, what they haven't shown is that it's any better than waiting until it grows and then zapping oligoprogressive disease. So, sometimes, I have to be honest, I do have some patients here who push for that. I'll actually get a PET scan, which is much easier for me on this side of the Atlantic, and just see if anything in the stuff is actually still lighting up on the PET scan. If it's inert, I sometimes say, your response is too good, there's nothing to zap, which is a good problem to have. But if there is somebody who's still, 3 months or so in, still has residual activity in a lump, maybe I'll zap it. I was involved in the original study of looking at that consolidation radiotherapy, and it's a neat idea, but in practicality, it's actually done relatively infrequently.
Okay, thank you. I've got another question, again, for patients who are on alectinib, because we obviously will have many. So this patient's written, I'm on alectinib and my bilirubin level has been 51 to 53 over the last few tests. What level of bilirubin is acceptable if other liver enzymes are normal? Professor Popat, do you want to start with that?
Yeah. Well, I mean, I remember.
Can someone tell me what the upper limit of normal is in the UK?
27.
Yeah.
Thank you.
Yeah.
Itch.
So I remember we did the one of the original alectinib phase 2 trials in the UK. I think there were only two centres. It was us and Aberdeen, and we enrolled the only patient to receive it. And the problem that we ran into is that his bilirubin went up, and it was beyond what the trial would allow. And so we quickly learnt that alectinib and bilirubin elevation is a problem, but it's usually not a problem for the patient because they're actually doing really, really well without any other liver problems whatsoever. And you have what's called this isolated bilirubin rise. We see that quite often with alectinib. It's probably something called Gilbert syndrome, where it's just a natural, normal variation of how the body copes with drugs, and it doesn't usually cause any harm, assuming that the rest of the other liver tests that we do on the blood tests don't show any other major problems. Now, if the bilirubin is elevated very high and the other liver enzymes are also elevated very high, then that's a different ball game, and that is a problem. But the common thing is to have a isolated bilirubin rise, and generally I tend to ignore it, to be honest with you, and just carry on. So I'm not sure I can give you an upper limit where I've been a bit worried. I guess I'd probably worry a bit at about 70, but I've a few that run between 30 and 50 all the time.
Yeah. Likewise, I've had a patient in exactly this situation. We've just ignored it. It's been very stable at about 55. So again, it had no clinical reason to change the alectinib, so we just carried on.
Okay, thank you. I think that'll be reassuring to whoever asked that question. So I've got another one here. So for Dr. MacKean, I started on crizotinib, then ceritinib, and have been on brigatinib for 18 months, having been told that this will provide better protection for the brain. In simple language, what is the blood-brain barrier? And if brigatinib crosses it, does it have adverse effects on the brain? I seem to have marked deterioration in my memory over the past year.
Oh, I'm sorry to hear that. That's not fun. And I would worry about that, and I'd hope that you've had a brain scan, and please ask for one if you haven't. It can affect the brain, so you're right to be worried, and you wonder about whether a dose reduction might be in order there. But the blood-brain barrier is the most amazing way your brain has of keeping out drugs that might cause the brain. Our brain was our most important thing when we were on the plains of Africa getting food for ourselves. We didn't want any drug to get in there, so it has a way of keeping it out, and drugs included can be kept away from the brain, but we don't want that when we're treating cancer. We want the drug to get inside the brain and treat the whole of your body from top to toe. And that's what brigatinib can do. So I would hope you can get some help with your memory. That's not good.
Okay, thank you very much. I'm going to change the direction of the question now for Dr. Camidge. Thinking of the work that's being carried out by Turning Point and Nuvalent, do you have any thoughts on the likelihood of fourth generation TKI being successful?
Yeah, so the first thing to point out is that the terms of generations of drug are, you can just make it up. There's no standard definition, and it's a little bit of marketing spin. So if Melanie, Sanjay, and I suddenly decided to cook something up in our garage, we can call it a 10th generation drug, and no one can stop us. What I think the key thing here is that both the Nuvalent drug and the Turning Point 0131 drug, they're working on a specific mechanism of acquired resistance, which is what's called compound mutation. So more than one resistance mutation piggybacked together. And the reason that's important is it's probably going to be very useful for those people, but it's not an answer for everybody. So you can't just assume that because they've developed a fourth generation drug, that's the drug for you. And I think what I really would like people to start to think about is to try and understand their mechanism of resistance, and then they can choose the best drug or combination of drugs based on that. But as we talked about in this prior meeting, one of the challenges in the UK is being able to rebiopsy and being able to have access to the kind of DNA sequencing tests to actually figure out the mechanisms of resistance. So I can throw that one back to Sanjay and Melanie.
Mm-hmm.
Yeah, I mean, I'll take that point about rebiopsying and sequencing, Ross. In England, we have a new NHS infrastructure for cancer gene sequencing, which is now funded. And in England, and Melanie, you can come in about Scotland, but in England at least, if we wanted to do a biopsy on a patient to do the sequencing for that tissue biopsy, we are now able to do that. Some centres have easier access to that than others, because whilst the NHS has said you can do it, not all centres have the ability to do it yet. But that is coming online, and that should be available. And as Ross has pointed out, the challenge sometimes is having the breadth of access to the different ALK inhibitors, because the NHS often doesn't give you enough choice for what you might want to do at what time you want to use it. So there are some limitations in what we can do, but gene sequencing is coming online, and I would be a strong proponent of, as Ross has said, trying to understand that that resistance mechanism so that we can put some science behind the decision making as to what the next steps might be. But Melanie, I don't know what the Scottish situation is.
It's looking better and better. We're certainly available in Glasgow. It's coming on stream in Edinburgh. But it's exactly that. We couldn't have envisaged this, a few years ago. So it's amazing. The speed of change has been absolutely brilliant. I think there was a lovely phrase Ross used earlier at the earlier meeting that I loved about a patient surfing the wave of discovery. And I think that is a beautiful phrase for patients to take on board of, we've got to try and get you on that wave and do our best to try and access these things for you.
Well, following on from that, Dr. Camidge, does the future lie with CRISPR gene editing?
I don't know why people always ask me about that. So the short answer is not anytime soon. So that's if the only way that's going to have a role in human beings at present is when there's something wrong with like all of the genes in your body. Let's say you have like a sickle cell disorder or some inherited disorder, and then you just need to edit it out. The problem with gene editing is, how do you get it to the cancer cells and not to your normal cells? And we're not there yet. So CRISPR is a technique which is awesome in the lab for messing around with DNA, but it is not ready to sort of stick into people. And for those of you who saw the slightly terrible Will Smith movie, The Day After Tomorrow, whatever it was, where everybody is turned into a zombie, I think that was done with CRISPR, and so we have to be slightly cautious about that.
Definitely. Okay. Professor Popat, the UK is involved with Project Orbis, which is a programme, for those that don't know, coordinated by the FDA in the US to review and approve promising cancer treatments. Will this reduce the timescale for regulatory approval in the UK?
I'm so pleased you asked me that question, because I've become a Project Orbis expert in the past 2 weeks. Because I think this is a really important step for the UK. In fact, almost everybody I speak to has never heard about it. So I'm delighted that you've brought this up. So Project Orbis is a mechanism by which a drug manufacturer, if they have a exciting drug which is classified as a breakthrough designation, i.e. it's a step change in the way we are treating a disease. It really is a big major step forward. It's a mechanism by which that drug manufacturer can submit their case for approval to the US regulator, that's the FDA, using this mechanism called Project Orbis. And at the same time, if the FDA approves it, then in parallel, a number of other global regulatory agencies have the ability to also approve it without re-reviewing all that data. And that includes the UK MHRA, because now post-Brexit, we are not completely governed by the European regulator, EMA. We're now governed by the UK regulator called the MHRA. And Project Orbis allows any approval that's gone down that route for a drug. If it's FDA approved, then the UK MHRA can also immediately approve it as well. Now, that doesn't mean to say that you can then use it within the NHS, because in the UK there's a second step, which is, once it's been approved, will the NHS pay for it? And that's something called reimbursement, and that's what a body called NICE does, at least for England, and I think Melanie will say something called SMC for Scotland. And so what that means is, if the FDA approve it, then immediately the MHRA are in a position to potentially approve it. And it then means that the NICE process, which gives the NHS the approval to use the drug in the NHS, has to kick in at the same time as the MHRA process, which is a lot earlier than it normally would. Now, we've never had a drug that's gone down this route before, so we don't really know how it might work. That's the theory. There is one drug, not for ALK-positive disease, but for another type of lung cancer, going down that route at the moment. And I, for one, am keeping a very beady eye on this, because it does have potential to be one of the good things out of Brexit, which is hopefully very rapid drug approval in parallel to the US FDA. But, you know, let's see where we go. It's, in theory, a great, great step forward.
Excellent. And does anybody else want to add anything to that?
Just to agree, I literally, first time I heard about it was earlier this evening, so I am learning fast, and I am super delighted to hear that, actually, that there is a mechanism in place, because it was one of the concerns we've had about leaving the EMA, the European Union, and that we'd be further down the tree for drug companies to submit for licensing. So we'll be really watching this space. Hope it works out. And yes, it's the SMC in Scotland that make the reimbursement decisions, but they can't begin to look at a drug until it's got its license. So the sooner it gets its license, the sooner it starts on that road. So brilliant.
Keep our fingers crossed for that one then.
Yeah.
Okay, lovely. Right. Professor Popat, is the ALK project national database in the UK used to inform research? And if so, how?
So the I presume, Deborah, this is talking about the data set that was captured by colleagues at Manchester at the Christie.
Yes.
Yeah. So I mean, that data has been published, and it's a retrospective review of how patients with ALK-positive disease were treated and what their outcomes were. And it was really used very much to benchmark how we were doing, because at the same time as Ross and colleagues in Colorado were publishing their experience, we were actually a little bit behind in our access to drugs and having to benchmark where we were in the UK. And I think it really demonstrated that we had a little bit of a way to go to catch up to some of the excellent work that's coming out from the US, and that's for multiple reasons. That data set, I don't think, is still being updated, although I stand to be corrected if Deborah, you know otherwise, and the audience knows otherwise. But there are a lot of discussions ongoing in the background within the UK about hosting what's called a molecular registry, where we really can document in a very systematic and organized manner the outcome of patients with what we call oncogene-addicted lung cancer. So this is lung cancer with a genetic alteration, both ALK, ROS, and there are a bunch of others as well, because it allows us really to understand what happens to our patients, why there is variation in care, why there are differences in access. And in the meeting we were just discussing, there are a number of open questions about what the right treatments are in certain conditions, and it allows us to map what happens to patients and their outcomes to then inform us, is this a treatment that we should really be doing, or is it really just giving us lots of side effects for our patients and really not a huge amount of benefit over other potential treatments? But I don't know, Melanie, do you want to comment further on that?
Yeah, I think this is where the NHS can really sing to a strength, that we can collect prospective data, and we need to do more of this. There's been the National Lung Cancer Audit, been running for many years now, and it's helped drive up standards. You know, places that were underperforming have then been questioned why they didn't meet, you know, the standard. So I think the more we can collect this data specific to ALK and the other oncogene drivers, it's going to really inform the future. So yes, please, let's have a prospective audit approach. That's one of the pleas, yes.
Thank you very much. Dr. Camidge, we read that you use tumour markers. Can you explain what they are and how you use them, please?
Yes, yes, I get a lot of stick over this one. So serum tumor markers are the things that are secreted by some cancers into the blood, and they have names. So most lung cancer hasn't read the textbook, so they spew out the markers that are associated with other cancers. So CEA, which is associated with colon cancer, CA125 with ovarian cancer, CA199 with pancreatic cancer, and CA2729 with breast cancer. And we did a study proving that the lung cancer hasn't read the textbooks, with something like 80 to 90+% of oncogene-addicted lung cancer express these markers. So what I use them for is I do not make a treatment decision on them. I don't say, I don't need to do a scan because look, your markers are going down or your markers are going up. What I use them for is to let me know when the imaging modality I'm using is missing something. So let me try and explain by that. Let's say you're being followed with CT scans and Dr. Popat is saying, Deborah, it looks fantastic. Your CT scan looks stable. You're still responding. But every time you're doing a CT scan in my clinic, I'd also be checking the blood markers. And if they started to go up, even if the radiology report said everything was stable, it would tell me something's going on. And then I have to do, at least here, I would do a PET scan because that's when I pick up a bone metastasis or a lymph node metastasis. And that's when I can go in with that oligo-progressive thing. So at least in our experience, the tumor markers are used to use PET scans in a cost-effective way and to try and pick up oligo progression more frequently.
Thank you very much. So following on from that, can I ask Sanjay and Melanie on your comments around tumour markers, please?
Yeah, I'll kick off. I mean, I think that's a very reasonable way of doing it. In the UK, I'll speak for England, we traditionally haven't been looking at CEA, but, we see this a lot, actually. And I see this a lot in patients that come across various parts of my practice from East Asia, where CEA monitoring is actually very, very common in patients with a different type of disease, EGFR mutant disease. And I think it can play a role where you have this sort of, I'm not really sure what's going on because the scan is telling me something else, but the patient's telling me something slightly differently. And it can help clarify the situation. So yeah, it can be used, but the other sort of counterpoint is that not all patients have a positive CEA in their tumor. So you sort of have to know what's going on at the beginning before you start any treatment to know whether it's worth following that up. Because if you're not producing CEA in your blood test at the beginning with your tumor, you're never going to produce CEA. So noting that it's fine and within the normal range every 3 to 4 months doesn't really tell you much. So there are little caveats with that, but it's not an unreasonable blood assay to do. The people in the GI world do it quite frequently, and the people in the gynae world do it frequently. We've not really done it in the lung world, and I'd be a bit hesitant to make a major treatment decision on the basis of the CEA only, as I think as Ross has explained as well. It all needs to be done in conjunction with all the other information that you have about that patient. But, Melanie, interested in your thoughts.
I agree. I've done gynae practice for 20 years. And the one caution I would have is it can produce an awful lot of anxiety, a blood test, people can get very fixated on one blood test and it's trends you're looking for. And sometimes, for instance, in ovary cancer, the difference between a blood test of 2,000 and 3,000 isn't as significant as a blood test between 20 and 23. And so it can be very complex to interpret these blood tests, and it can lead to a lot of anxiety. So just a word of caution, if you're using them in a particular way, that's great. But if you're using them as reassurance, sometimes it can offer false reassurance, and sometimes it doesn't tell you exactly when to change treatment on one blood test. Okay.
So if there was a situation, as Dr. Camidge has just described, that someone's CT scans look fine, but the patient is telling you something, that they're not feeling fine, our tumour marker testing, is it available in the UK on the NHS?
Yep.
Would it be reasonable for a patient to then ask that this is looked at?
I don't think it's unreasonable, but it needs to be, the results need to be interpreted with the whole of the picture as to what's going on with that patient. Yep. And it's it's usually not the tumor marker or the CEA level itself which by itself is giving you the diagnosis of what's going on or helping you. It's everything in combination that really gives you the whole picture.
Okay, thank you very much. Dr. MacKean, it's another question for you. All oncologists carry out regular scans of the body. They don't wait for symptoms, but some won't carry out brain scans until there are symptoms. We can't understand the logic of this. We know of the driving license implication, but that is a matter for the patient to consider. And it's saying here, not the oncologist. What are your thoughts?
I have to say I've gone full circle on this one, from being somebody who didn't want to do scans for partly because of the driving license thing, to coming around to thinking that this is an important part of an ALK patient's journey, is to do regular scans. We struggle to get MRI scans in Scotland, and that's an access issue. But if we're worried about a CT scan, we can certainly request an MRI scan. I don't know what the situation is, Sanjay, for you guys.
Yeah, thanks. I think, Deborah, part of the problem that people perceive in the UK is just lack of access to MRI scanning capacity in the UK. There just isn't enough UK MRI scanning capacity that you and I would want. I mean, for the record, I'm a believer in scans. I mean, I think you know that anyway, with what we've said in support of the ALK positive booklet that you've written, supporting regular brain scans. And part of that is driven by what you can do with that information. I mean, bearing in mind, let's ignore the driving issue. Now, if we didn't have any other treatments, then you know what's the point of looking? But now we do have treatments. We have highly effective ALK inhibitors, but more importantly, we have availability of SRS, or stereotactic radiosurgery, where we can potentially pick off disease which is growing and recurring to allow patients to continue on their ALK inhibitor for longer. And the more we're using this technology over the past 5 to 10 years, we're learning about it, and we're becoming more aggressive and using more of the technology. So in part because we have options now, then we can do it more. And I definitely think it's the right way forward. But undoubtedly, unfortunately, there are many places in the UK where access to MRI scans of the brain is just really very limited because of trust issues. It's not helped, I have to say, by the fact that NICE do not recommend brain scanning on patients with stage 4 disease, because they have not felt that it was cost effective for whatever reason, and I don't really understand or support their decision in that. So until NICE really changes its views, then the hospitals which have problems really don't have much to say, because NICE is saying that you shouldn't really be doing this. So that's one of the reasons why I'm very supportive of the work that your group has done, Deborah, in promoting brain MRIs, because it is that additional advocacy work which can help pressure to make scans a reality for some of our patients.
Okay, thank you. I've got a question again for Dr. MacKean, but actually I would also value Ross Camidge's response. How can patient groups influence the amount of research that is done in the UK? And are you aware of any trials in the UK that are relevant to ALK patients?
I will probably defer to Sanjay for this one, but I think you can have a huge role to play, because talking to each other, you can start to hear of things that are going on. And I think we are learning more and more, and I've had more and more patients who have gone from one thing to another. And that is the paradigm with ALK, is multiple treatments that do well. So we need to support that research, but I would defer to Sanjay, who's a much more active researcher than myself.
Yeah, thanks, Melanie. So I think patients are absolutely critical in research, because actually it's the patient questions that patients have which drive our progress. And I think there are really two basic types of research that we do in the UK, and these are coordinated by a UK government organisation called the National Cancer Research Institute, which coordinates clinical research in the UK. So we have research which is looking at new drugs. And that is really welcome because we want to make step changes. We want to really advance the field. These sorts of research examples are things like clinical trials when we're using new drugs, and these are run by pharma companies, and they will be usually, because of various issues, they'll be doing their early tests in human beings, usually in the US, and then when they've worked out that this drug is viable, it's not just a chemical, it might just really be a drug, then it might then globalized to be tested in small numbers of patients in the rest of the world. And the UK is really keen to do those types of studies. So in terms of what drug trials we've got on at the moment in the UK, there is one drug trial which is not run by a pharma company, but is run by an academic group. So an academic group is a group of consultants that want to do research, and they've sort of got together around a coffee table and said, look, we want to investigate this particular area. And in the UK at the moment, there is a trial looking at lorlatinib and looking at something called liquid biopsies with lorlatinib, which is run by a European / UK group called the EORTC, and that's open in a couple of centres within the UK. But in terms of drug development, we are very much wanting to develop new drugs in the ALK space, and we're very much looking forward to the Turning Point Therapeutics new drug. As soon as Ross has worked up the right dose and the schedule, we want to get it straight across. And your members can rest assured that the likes of us and others are having those discussions with Turning Point Therapeutics, with the company, on a regular basis to ensure that when the drug is ready to come to the UK, we're happy to receive it. But your question was sort of broader about what patients can do. So of course they can look out for new drugs, which is useful for their particular condition, but influencing the scope of research is a really critical area, and we're really looking forward to interacting with patients and patients advocates. So for example, the UK NCRI have a one-day workshop on never smoking lung cancer coming up in the next couple of weeks. And that workshop is a whole day workshop aiming to just chat through some of the scientific issues around why people who never smoke get lung cancer, what happens to them, what are the open research questions that we have to answer to understand the disease better, and then trying to formulate a national strategy for what sort of research we can do in this field. And none of that research is going to be possible if it's not supported by input from patients and patient organizations. So what can patients and patient organizations do? You can really help us in trying to work out what the key questions are, because hey, I think I might know, but maybe I haven't got a clue. And actually what's really important is something that's completely different. And so I'm all ears. I want to know everything about what's important.
And just for your team, the NCRI website is very good to look at if you're ever wanting to look up, you've got to that stage, you want to have a look at research trials. That's the website I would go to.
Thank you. Ross, have you got anything to add?
No, apart from the fact it's actually been a wonderful journey, because most of the folk on here knows that lung cancer wasn't breast cancer. We didn't have thousands of people marching, and now we do have survivors and thrivers, and they're starting to become involved. So at least in the US, I'd say there's 4 different things that they get involved with. So they can raise money, so philanthropy is big over here. They can lobby, and I think this is incredibly important. We were talking in the last meeting about, here are the national guidelines as to what you should do. Well, where's the patient advocate on there saying, well, that's really dumb? The idea about pushing for brain scans, the idea about, why are you saying I should do this or should do that? So I think advocating on the things that are important. I think contributing tissue, this sounds like a bizarre thing, but Bonnie Adaria, who's one of the advocates over here, says the answer is in ourselves. So when you progress, if there actually had a UK-based thing where you could rebiopsy and could reanalyze that, they get to ask your permission, so you get to contribute to that knowledge. And then the fourth one is, you do get to contribute to research in a much broader way. And I touched on this in the last thing, that we've actually, for example, many ALK positive patients are young women who are at the time of their life when they're thinking about having babies. There is zero collated data in terms of how these people do. So we literally a few weeks ago started an international database just to try and find out how people have done. You know, did they have a termination? Did they carry the baby? Did they decide to use a surrogate? It's incredibly emotive. We do have to fact check everything that goes on. But if we don't start to collect that data, we'll never know. And that data is with you guys.
Okay, thank you very much indeed. We are very close to 9 o'clock. Can't believe how quickly the time has gone. So we've got maybe a controversial one here that we might be finishing on. I'll give all three of you the opportunity to answer. The UK and the US have very different healthcare systems. Which produces the better outcomes?
Oh.
I thought you'd like that one.
So do you want to know the short answer? It depends on how much money you've got. So if you've got lots of money, the US does. If you don't, or you want someone else to pay for it, the UK is much better.
I have to say, Ross, I once looked at the survival figures for Black Afro-Americans, and it was not funny with lung cancer. Yeah. It was a lot worse than Scotland. So it depends who you are in America. Yeah. Unfortunately, that's the easy answer.
I think that's right. I mean, I think at the end of the day, we whinge a lot about the NHS, but actually, if you look at what's available, how much it costs, and the fact that the NHS is providing it free of charge, without any cost, it's actually quite remarkable. So, pros and cons on both, I think.
Yeah. I mean, I like to think of it. So the UK is a fixed price menu, but nobody starves.
Yeah.
Yeah.
Good way to think about it. Right. Okay. We've got just 2 minutes, so I'm going to put one last question to you. That's just your thoughts on. Obviously, over the last 12 months with the pandemic, the ways of working has changed considerably, especially for yourselves with virtual consultations. Do you see face-to-face consultations ever returning to the old normal?
I hope so.
Yes, please.
Yeah, I mean, I have to say, virtual consults are great if they're a brief, everything's fine, pat you on the back, and off you go. We meet again in 2 or 3 months' time. But actually, if there's anything complex to discuss, it's much better to do. Personally, I find it's easier to do things face to face because you can just tell what's going on with people face to face. Just looking in the eyeballs is just part of what we do as physicians. That's what we're brought up, what we're trained to do. It's what we've been doing for years and years and years. And I personally, don't think it's very easy to do on a flat screen, but welcome other folks' comments.
Oh, on the plus side, I have got to meet a lot of pets. And I think there's a theory here that actually, on the plus side, if you are in your own home and you have your dog, your cat, and even if it is not such good news, sometimes that can be very comforting. So there's a plus side to everything. I think as doctors, we're super uncomfortable because we haven't been trained that way. And I miss what I call the forearm squeeze. I miss the look in the eyes. I miss that non-verbal chitter-chatter coming into the room, watching people walk into the room. I miss all of that because I was trained that way. I guess if I'd had the training, I might feel a little bit more comfortable about remote consultations, but...
Guys,
and the last word from you.
I'm going to have to jump. Deborah, I'm going to have to jump off. I'm so sorry.
That's okay. We've just come to the end anyway. So, lovely. Thank you very much indeed, Dr. Camidge. Thank you. I'd also like to thank both of you, Dr. MacKean and Professor Popat, for your time this evening. It must have been an incredibly busy day for you guys, having already attended one meeting to then at 9 o'clock at night still being on another meeting. So I won't hold you up any longer. I think you deserve to now sign off and go and relax. But on behalf of all our members, I can't thank you enough for sparing the time to speak with us this evening and for answering our questions. Thank you very much indeed.
You're welcome. Take care.
All the best.
Thank you.
Bye-bye.
Bye-bye.
Bye-bye.